Hello students, hello everyone, good morning all. So I've launched the technical poll, let me know, okay, my screen is visible and my audio is clear. So today we are going to discuss cell cycle and cell division. As you all know that cell cycle are the sequential events and they are the coordinated events, right? Good morning, MS Gauri. So the first thing that we are going to discuss is the phases of cell cycle. First thing is the duration of cell cycle. The duration of cell cycle vary from organism to organism. So there's a variation in duration. Okay, the duration of the cell cycle vary. In yeast, it is 90 minutes. In humans, it is 24 hours, where the 1 hour is for the nuclear division or the cell division, and 95% of the cell cycle duration is occupied by interphase. Cell cycle is majorly divided into two phases. What are those phases? Number one is the interphase. And the second one is the M phase. Okay, interphase further consists of G1 phase, which is the growth phase, S phase, where duplication of DNA and centrosome takes place, and the third one is G2 phase, where the synthesis of protein takes place, which are required for the M phase. And M phase is the division phase. Okay, and the M phase is consists of karyokinesis and cytokinesis. Karyokinesis is the nuclear division and cytokinesis is cytoplasm. When cytoplasm gets divided, it is known as cytokinesis. Karyo always means nuclear, okay? Cyto is referred to the cytoplasm. Now moving to next one, the interphase. As I told you that interphase has G1 phase. G1 phase is the growth phase. Where the cell is metabolically active and it continuously growing. S phase is where the DNA duplication takes place, where the content of DNA doubles. And in the S phase also, duplication of centrosome takes place. Remember this, in G2 phase, the protein are synthesized which are required for M phase, the mitosis phase. In G0 phase, which is the quiescent phase or the also known as the resting phase, where the cell do not divide and it enters into the inactive stage, like your neurons, heart cells. Okay. Moving to next is mitosis. Mitosis, we call it as equational division because the amount of the chromosome is constant throughout the generation. Okay, so the mitosis is divided into four phases. Number one is your prophase. Number two is the metaphase. Number three is anaphase. Number fourth is telophase. First, we are going to discuss prophase. So prophase, there are few characteristics which are related to prophase. Number one is the then nucleolus, nuclear membrane, Golgi bodies, and endoplasmic reticulum disappear. So the cell organelles get disappear in prophase. Disappearing of nuclear membrane get disappear, nucleolus get disappear, cell organelles get disappear in prophase. And the condensation of chromatin start. The chromatin start condensing and result in the formation of chromosome. Centrosome, which begins to move towards the opposite pole. The centrosome, which get duplicated in the S phase, start moving towards the opposite poles in the prophase. Remember this, very important point. Can see this fragments of, these are the fragments of nuclear envelope, because the nuclear membrane, it does start disintegrating. Okay, disappearing of nuclear membrane occurs in prophase. Moving to next one is the metaphase. So in metaphase, the morphology of chromosome is easily studied. Okay, the morphology of the chromosome is easily studied in metaphase. Remember this point. Spindle fibers, they get attached to the kinetochore of the chromosome. Let me draw. This is the chromosome. Here is the disc-like structure. This disc-like structure are known as kinetochore, where the spindle fibers attached like this. These are the spindle fiber, they get attached. Okay, and the chromosome, they move to the equator, get aligned along this metaphase plate. This is the metaphase plate. So all the chromosome, they get aligned at the equator, and the spindle fiber, they get attached to the kinetochore of the chromosome. Moving to next is the anaphase. In anaphase, the centromere split and the chromatids separate. And see these? The centromere split and the chromatid, they get separate. They start moving towards the opposite pole like this. They start moving. One is moving to this pole, the other one is moving to this pole, and the chromatid, they move to the opposite pole. Remember, the anaphase feature is the division of the centromere, and the sister chromatids get separated. In mitosis, the sister chromatids get separated. Remember this point. Okay, now moving to next one is the telophase. So in telophase, it is the last stage of the mitosis, and in telophase, the nucleolus, nuclear membrane, they start reappearing. You can see these? These are the nuclear membrane. They start reappearing in the telophase. And the chromosome start decondensing. In prophase, the chromatin material start condensing to form the chromosome in prophase, but in telophase, now the chromosome will start decondensing to form chromatin. Okay, this is the first point, says the chromosome cluster at the opposite spindle pole, their identity is lost as discrete element. They start forming chromatin. Nuclear envelope develops around the chromosome. I told you that nuclear membrane start reappearing in the telophase. Nucleolus, Golgi, ER, they start reappearing in the telophase stage. Okay, now this is all about the karyokinesis. Next moves to cytokinesis. So cytokinesis is referred to the division of cytoplasm. When the cytoplasm divides, and from the one parent cell, we get the two daughter cells. Here, the cytoplasm divides. So in case of animal, cleavage furrow is formed. And the formation of cleavage furrow is centripetal. Centripetal is from the periphery towards the center. You can see this furrow? It's moving from the periphery towards the center. Okay? In plant cell plate formation occur. And the formation of the cell plate is centrifugal. That is from the center towards the periphery. First, it will form in the center, and then it will start move towards the periphery. So the formation is centrifugal. Okay. Remember one point is phragmoplast. During the cell plate formation, some remains of the spindle fibers and the secretion by the Golgi body, they play a very important role in the cell plate formation. Okay. If the karyokinesis, that is the division of nuclear, is not followed by cytokinesis, is not followed by the cytoplasm division, it results in the nucleate condition. Okay. If the cytoplasm does not divide and the one cell is containing a many nucleus, then its condition is known as multinucleate condition. We also called it as syncytium. Okay, you can see this in liquid endosperm in coconut. Remember this example, because NEET love examples, exceptions, right? So remember all these exceptions or the examples which are given in NCERT. Importance of mitosis. As I told you, in mitosis, if this is a parent cell, this parent cell will divide into the two daughter cells. This is daughter cell number 1. This is 2. Okay. So the genetic material in the parent cell, the daughter cell will also have the same genetic material. They will be identical, or we call it clones. In mitosis, we get clones. Clones are those which are morphologically and genetically identical to their parent cell. So in mitosis, we get the daughter cells, they contain identical genetic complements. Growth of multicellular organism. So growth in multicellular organism occur due to mitosis. Restore nucleocytoplasmic ratio, very important. When the cells divide, sometimes the nucleocytoplasmic ratio of the cell get disturbed, or the surface area to volume ratio get disturbed. So to restore this ratio, the cell has to further divide again to restore this ratio. So mitosis play a very important role in nucleocytoplasmic ratio, in restoring this ratio. Cell repair. Mitosis also play a very important role in cell repair. Okay, when we like if you have any injury, if you have any cut, right, the epidermis cells, they start dividing to repair that tissue. Okay, so there's mitotic division takes place there. Mitotic division in the meristematic tissue. Like plants, they have a root apical meristem, they have a shoot apical meristem. Okay, and that meristem, they show a continuous cell division because that cell is continuously dividing. So there the division is mitotic division. Moving to next is the meiosis. So meiosis involves two sequential cycles of nuclear division and cell division, that is meiosis 1 and meiosis 2. Okay, and the gap between the meiosis 1 and meiosis 2 is interkinesis. And it is very short, for very short duration. But there's only a single DNA replication. Okay? Remember this point, beta, it's very important that meiosis involves two sequential cycle of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. There will be a single cycle of DNA replication. Meiosis involves recombination. In mitosis, there is no recombination. There is no crossing over. Okay? But in meiosis, it involves recombination. And recombination leads to variations, like genetic variations. And these variation leads to evolution. If the cell is having a variation or the genetic variation, that means that the organism can adapt to the adverse condition of the environment. Okay, that will lead to evolution. The gradual changes in an organism, it leads to evolution. So those gradual changes are the genetic variations. End product is 4 haploid cell. In mitosis, we get two haploid. In mitosis, we get generally 2 cells, right? One parental cell, it's dividing and it forming the 2 daughter cells, which are identical to their parent cell. But in meiosis, we are getting the 4 haploid cells. Remember this point, very important. The end product is 4 haploid cells. Remember the microsporogenesis in plant, where the microspore mother cell, it undergo meiosis and the 4 haploid microspores are formed. Right? Similarly, the megasporogenesis, where the megaspore mother cells divide mitotically and the 4 haploid megaspores are formed, right? So the end product is the 4 haploid cells, and the meiosis occur in the gametic cells. Remember this point. Mitosis occurs in somatic cells and meiosis occurs in gametic cells. Moving to the next one, the meiosis 1. Meiosis 1 has 4 stages, that prophase 1, then metaphase 1, anaphase 1, and telophase 1. Okay, so prophase 1 is more complex and longer. Remember this, prophase 1 of meiosis 1 is longer and more complex. Why? Because it contains further stages. There's leptotene, there's zygotene, there's pachytene, there's diplotin, and there's dikinesis. So the prophase 1 is consist of 5 stages. Number 1 is the leptotene stage. In leptotene stage, what happens? The chromosome becomes gradually visible, and the compaction of chromosome continues throughout the leptotene stage. The compaction of chromosome, it starts in the leptotene stage. In zygotene stage, synapses, very important point, beta, synapses. Synapses, very important. Synapses is the pairing of chromosome. When the homologous chromosome they pair, that process is synapses, and the complex form is known as synaptonymal complex. And the paired homologous chromosomes are also known as bivalent or tetrad. See this? For more clarity, I'm drawing it. Okay, the pairing is taking place between the homologous chromosomes. And that pairing, the pairing of homologous chromosomes are known as synapses. Synapses is a characteristics of which stage? Zygotene. Remember these points, beta. They are very important. And the complex form is synaptonymal complex. And the paired synapse homologous chromosome is bivalent or tetrad. Bivalent consists of pair of homologous chromosomes. So it's bivalent. Tetrad because it consists of 4 sister chromatids. 4 chromatids are there. Moving to next is the pachytene stage. Pachytene, the characteristic of pachytene stage is recombination, crossing over. In mitosis, there was no crossing over. Remember this point. In mitosis, there's no crossing over because mitotic division are taking place in somatic cells only. In pachytene, the recombination is there. Recombination is crossing over. between the non-sister chromatid of homologous chromosome. Remember this point, very important. Crossing over takes place between the non-sister chromatid of homologous chromosome. And this recombination is catalyzed by a enzyme, and that enzyme is recombinase. Okay, and the bivalents in tetrads are clearly visible in Pachytene stage. They are forming in zygotene stage, but they are clearly visible in Pachytene stage. Okay, in this figure you can see this. There's a crossing over has taken place. This is the blue part and this is the pink part. Exchange of genetic material has taken place between the non-sister chromatid of the homologous chromosome. Next is the diplotin stage. In diplotin stage, dissolution of synaptonemal complex. Remember the zygotene stage, the synaptonemal complex is formed between the two homologous chromosomes. You know? Just right now I told you. This is that synaptonemal complex or the synapses. But this synapses or the synaptonemal complex get dissolved in diplotin stage. This get dissolved. It will get dissolved. But the chromosome, they remain attached at one point. And what is that point? Is the recombination nodule, the point where the recombination has taken place, the point where the recombination nodules are formed. At that point, the chromosome, they remain attached. Though the cytonymal complex has dissolved, but the chromosome are still attached at the point where the recombination has taken place, and they form, because of that they form a X-shaped like structure, and that X-shaped like structure is known as chiasmata. Call it at chiasmata. So chiasmata formation occurs in diplotin stage. Moving to next is dikinesis. In dikinesis, terminalization of chiasmata. Now so dikinesis, this stage is characterized by terminalization of chiasmata. In dikinesis, the chromosome are fully condensed. Mitotic spindles are assembled. Nucleolus disappear. Nuclear envelope breaks down. These all are the characteristics of dikinesis. Remember the characteristics of the stages, beta. It's very important. Sometimes you might get a question in like match the followings. Okay, or you can ask about like what is the mismatch pair. They can give you in list one, they can give you the name of the stages. In the list two, they can give you the features or the characteristics of each stages. So it's very important to remember the characteristics of each stage. It's very important. And these stages, 5 stages of meiosis ones are very important. Remember all these points. See this, the nuclear membrane is disappearing in dikinesis. Nucleolus get disappear in dikinesis. Meiotic spindles is assembled. Moving to next one is a metaphase 1. So after prophase 1, the next stage that comes is the metaphase 1. In metaphase 1, the bivalent chromosomes align on the equatorial plate. The paired homologous chromosome. Remember this point, very important. In meiosis 1, the paired homologous chromosome. The paired homologous chromosome, they get aligned at the equatorial plate in metaphase 1 of meiosis 1. You can see these paired homologous chromosome, they're aligning at the equatorial plate. And the spindle fiber, they attach to the kinetochore of the homologous chromosome. The spindle fiber from this point is attaching to this kinetochore, and the spindle fiber from this centrosome is attaching to this kinetochore. Okay. Next one is the anaphase 1. In anaphase 1, the homologous chromosomes, they separate. But the sister chromatid remain associated at the centromere. Remember this, it's very important point. Homologous chromosomes, they get separated, they start moving towards the opposite pole in anaphase 1, but the sister chromatid, they remain associated at their centromere. The sister chromatids, they're still associated with the centromere, but the homologous chromosomes, they get separated, they start moving towards their opposite poles. It's very important. Remember this, beta. It's very, very important. Moving to next one is the telophase 1. In telophase 1, the nuclear membrane and the nucleolus reappear, which get disappeared during the dikinesis stage of prophase 1 of meiosis 1. It gets reappeared in the telophase 1 of meiosis 1. Can see this nuclear membrane, it's reappearing. Similarly, all the nucleolus or the cell organelles, they will also reappear in the telophase 1. And telophase 1 is followed by cytokinesis 1. Okay, so telophase 1 is followed by cytokinesis 1, and this dyad is formed. First, the parent cell is dividing into the first in meiosis 1. This is meiosis 1. 2 daughter cells are formed. These will further go into division. Now 4. Now you will see 4 daughter cells, 4 haploid daughter cells will form. So we have completed till meiosis 1. Let's move to meiosis 2. In meiosis 2 is, first stage is the prophase 2. As I told you that gap between the meiosis 1 and the meiosis 2 is interkinesis, and that interkinesis is for a very short duration. So immediately after the interkinesis, the prophase 2 begins. In prophase 2, the nuclear membrane disappears. Again, these are the characteristics of prophase, like the nuclear membrane get disappear, the chromosome, they start becoming compact, they start becoming condensed. Okay? The cell organelles, they get disappear in prophase 2 as well. This nuclear membrane, they start undergoing disintegration. Moving to next is the metaphase 2. In metaphase 2, the chromosome aligned at the equator. Microtubules from the opposite pole of the spindle get attached to the kinetochore of sister chromatids. So in metaphase 2, what will happen? So remember in metaphase 1, the paired homologous chromosome, they were aligning at the equator, right? But in metaphase 2, the chromosome, in metaphase 1, they were paired. Remember this point. In metaphase 1, they were paired homologous chromosome. But in metaphase 2, the chromosome align at the equator. And the microtubule from the opposite pole of the spindle, they get attached to the kinetochore of the sister chromatids. See this? These are the spindle fiber. They are attaching to the kinetochore of the chromosome. Similarly, in this cell also. Next one is the anaphase 2. In anaphase 2, the splitting of centromere takes place. And because of the splitting of centromere, the sister chromatid gets separated and they start moving towards the opposite poles, right? Whereas in anaphase 1, the paired homologous chromosome, they get separated and they start moving towards the opposite pole, right? But in anaphase 2, the sister chromatids, they start moving towards the opposite pole. So meiosis 2 is same as the mitosis. That is why we call meiosis 1 is the reductional division and meiosis 2 as the equational division. And how these separation takes place, like how these sister chromatid gets separated. Number one, the splitting of the centromere of each chromosome. Number two is the shortening of microtubules. These microtubules, you can see these microtubules or the spindle fiber, they get shortened. Okay. And because of that shortening, the chromatid, sister chromatid, they start moving towards the opposite poles. Next one, after anaphase 2, it is after anaphase 2, it is telophase 2. The two groups of chromosome once again get enclosed by a nuclear envelope. The nuclear envelope which disappear in the prophase 2 reappear in the telophase 2. Nucleolus, other cell organelles, they get reappeared in the telophase 2. And the decondensation of the chromosome occur. Now the chromosome will convert into chromatin. Remember all these points, beta. These are very important. Telophase 2 is followed by cytokinesis 2, where the 4 haploid daughter cells are formed. The end product of meiosis 2 are the 4 haploid daughter cells. Remember this point, very important. You can see this. After cytoplasm division, this is 1, 2, 3, and 4. 4 haploid daughter cells will be formed. Moving to next is the importance of meiosis. First, very, very important point regarding meiosis is the recombination. I told you that recombination occurs in the Pachytene stage of prophase 1 of meiosis 1. So during recombination, genetic changes, exchange of genetic material takes place. And because of this exchange of genetic material, variation occurs. And these variation, they leads to evolution. Very important point. So we have completed the cell cycle, main points regarding the cell cycle and the cell division. Now we are moving with the questions from the previous year. Please let me know in the chat box if you have any doubt regarding cell cycle and cell division, which I have covered so far. All right, then we are moving with the question number 1, which says, which of the following never occurs during mitotic cell division? Option 1 says movement of centriole towards opposite pole. Option 2 says pairing of homologous chromosome. Option C says coiling and condensation of chromatin. Option 4 says spindle fibers attached to kinetochore of chromosome. What do you think? What will be the correct option for this question? Students, everybody. MS Gauri, Ritu, Lakshmi, Chandana. What do you think? What will be the correct option for this question? Asha, Deepak, Shreya. What do you think? What will be the correct option for this question? They're asking about which of the following never occur during the mitotic cell division. Just now I told you, like all the stages of mitosis, there's prophase, there's metaphase, there's anaphase, there's telophase. What do you think? Okay, Shreya says option B. Okay. Option 1 says the movement of centriole towards opposite pole. Yes, in mitosis, the centriole, they move towards the opposite pole in prophase, right? Option 2 says pairing of homologous chromosome. This is the correct option. Pairing of homologous chromosome is a characteristics of meiosis. I told you many times that in meiosis, the pairing of homologous chromosome occur during the zygotine stage of prophase 1 of meiosis 1. Remember this point, beta. It's very important. Question 3 says coiling and condensation of chromatids. Yes. In prophase, the coiling and condensation takes place, right, to form the chromosome. Fourth option says spindle fiber attached to the kinetochore of chromosome. Yes, true. In metaphase, the spindle fibers from the opposite pole, they attach to the kinetochore of the chromosome. So all the options are correct except this two. Option number 2, which says pairing of homologous chromosome. It is a characteristic of meiosis, not mitosis. And the question is asking about mitosis. Very good, Shreya. Moving to the next one. Which is incorrect statement about meiosis? This question came in NEET 2021. Option 1 says meiosis involves two sequential cycle of nuclear and cell division called meiosis 1 and meiosis 2, but only one single cycle of DNA replication. Option 2 says meiosis 1 is initiated after the parental chromosome have replicated to produce sister chromatid at the S phase. Option 3 says meiosis involves pairing of non-homologous chromosome and recombination between them. Option 4 says haploid cells are formed at the end of meiosis 2. What do you think? What will be the correct option for this question? It's a very easy question. Try to remember all those points which I have just told you. Shreya says option 4. Shreya, they're asking about the incorrect statement about meiosis. MS Gauri says 3. What about others? Asha, Ritu, Chandana, Deepak, what do you think? What will be the correct option for this question? Lakshmi says option B. Always pay attention to this, whether the question is asking about the incorrect statements or the correct statement. The question says which is incorrect? Which is incorrect statement about meiosis? Incorrect statement. Meiosis involves two sequential cycle. Yes. Nuclear cycle and cell division. Yes. Meiosis, we call it as meiosis 1 and meiosis 2. Correct. Only a single cycle of DNA replication. Yes, there's only a single cycle of DNA replication. The DNA is duplicated only once, right, in meiosis. So this is a correct statement. Option 2 says meiosis 1 is initiated after the parental chromosome have replicated to produce sister chromatid at the S phase. Meiosis 1 is initiated after the parental chromosome has replicated to produce sister chromatid at the S phase. Is absolutely true. Once the parental chromosome has replicated to produce a sister chromatid, the S phase is followed by the G2 phase, and after the G2 phase, immediately there is a meiosis 1. So this is also a correct statement. Statement number 3 says meiosis involves pairing of non-homologous chromosome and the recombination between them. With the non-homologous, I told you in meiosis, in zygotine, pairing occur between the homologous chromosome, not between the non-homologous chromosome. And meiosis involves, in option 3 says, given that meiosis involves pairing of non-homologous chromosome. This is a wrong statement. And the question is asking which is the incorrect statement. So our correct option with respect to question is it's option number 3. Okay. And option 4 says 4 haploid cells are formed at the end of meiosis 2. Yes, it is true. Many times I told you that the end product of meiosis is the 4 haploid cells. Now it is clear. Shreya, now it is clear. Very good, Lakshmi. Very good, MS Gauri. You have answered it correctly. Moving to the next one, the oocyte of some vertebrate get arrested for years or months in dash. Number 1 is a diplotin, number 2 is dikinesis, number 3 is metaphase 1, number 4 is telophase 1. What do you think? What will be the correct option for this question? It's a very easy question. It's a straight line question from NCERT. The oocytes of some vertebrates get arrested for years or months in which stage? Name that stage. What do you think? What will be the correct option for this question? The oocyte of some vertebrates get arrested for years or months in dash. Which stage? Name that stage. Yeah, it is diplotin stage. The oocytes of some vertebrate, they get arrested in the diplotin stage. In dikinesis, what is the characteristics of dikinesis? The nucleolus get disappear, the nuclear membrane get disappear, right? Condensation of the chromatids occurs in dikinesis. In metaphase 1, what happened? The paired homologous chromosome, they get aligned at the equatorial plate or the metaphase plate, right? Option 4 says telophase 1. What happened in telophase 1? The nuclear membrane which get disappear in the prophase 1 reappear, nucleolus reappear, cell organelles reappear, right? And the chromosome, they start decondensing to form chromatins. I hope this is clear. Moving to the next one, this question came in NEET 2022, which says select the incorrect statement with reference to mitosis. Now you have to select the incorrect statement. Pay attention to this, whether the question is asking about the correct and the incorrect statement. Here, the question is asking about the select the incorrect statement with reference to mitosis. Option 1 says the spindle fiber attached to centromere of chromosome. Option 2 says chromosome decondense at telophase. Option 3 says splitting of centromeres occurs at anaphase. Option 4 says all the chromosome line at the equator at metaphase. What do you think? What will be the correct option for this question? They're asking about the incorrect statement. Okay, Deepak says 1. Ritu, Chandana, what do you think? What will be the correct option for this question? They're asking about the incorrect statement regarding mitosis. Ritu says option 3. Okay. Chandana, what do you think? What will be the correct answer for this question, beta? Whether it's option 1, 2, 3, 4. Okay. So the question is saying select the incorrect statement with reference to mitosis. Option 1 says spindle fiber attached to centromere of chromosome many times. Shreya says option 3. Say spindle fiber attached to centromere of chromosome. Spindle fiber attached to centromere of chromosome, beta. Is it a correct statement or is it is an incorrect statement? It is an incorrect statement. Centromere, spindle fiber, they attach to kinetochore. Many times I've told you that spindle fibers are attached to kinetochore of chromosomes. Option 2 says chromosome decondense at telophase. Yes, chromosome, they condense at prophase but decondense at telophase. Option 3 says splitting of centromeres occur at anaphase. It is splitting of centromeres occurs at anaphase. Because of that splitting of centromere, the sister chromatid gets separated and they start moving towards the opposite pole. This is a characteristic of anaphase. Shreya, I hope this is clear now. Fourth one is all the chromosome lie at the equator at anaphase. Very true. All the chromosome, they get aligned at the equator or the metaphase plate in metaphase of mitosis. So all the options are correct except the first one. Select the incorrect statement with reference to mitosis. Spindle fiber attached to centromere of chromosome is the incorrect statement. Moving to the next one. So the following figure represent. There's a figure is given in the question and they're asking about which figure, which option represent this figure, whether it's option 1, which is metaphase, option 2, transition to metaphase, option 3 is anaphase, option 4 is telophase. What do you think? What will be the correct option for this question? Ritu says 4. Ritu? Veda, can you see the nuclear membrane? There's no nuclear membrane. I told you telophase, the characteristic of telophase is the presence of nuclear membrane. They're reappearing in telophase. In this figure, you can't see any nuclear membrane. MS Gauri says 3. Shreya says 3. Okay. So in metaphase, what happened? All the chromosomes, they get aligned at the equatorial plate, right? This is the equatorial plate. All the chromosomes, they get aligned like this. I'm drawing only 2 chromosomes, okay? And these are the spindle fiber. So this figure or this figure, are they same? No, right? So it is not metaphase, because in metaphase, the chromosome, they get aligned at the equatorial plate. Deepak says option 3. Anaphase. Very good, Deepak. Second option says transition to metaphase. No, beta, this figure does not represent the transition to metaphase. Option 3 says anaphase. Yes, very important. Anaphase. The centromere, they split and the sister chromatid gets separated and start moving towards the opposite poles. Right? So this figure represents the anaphase. Telophase, no. Because in telophase, the nuclear membrane, they get reappear, nucleolus reappear, cell organelles reappear, and the chromosome, they become decondensed. So this figure represents anaphase. Question number 6 says, which of the following stages of meiosis involves the division of centromere? Which of the following stages of meiosis represent or involve the division of centromeres? Number 1 is metaphase 1. Option 2 is metaphase 2. Option 3 is anaphase 2. Option 4 is telophase 2. What do you think? What will be the correct option for this question? Now all of you can answer this. It's very easy. Involves the division of centromere. Deepak says option 3. Very good, Deepak. Yes. Which of the following stages of meiosis involves division of centromere? When the centromere divides, the sister chromatid gets separated and they start moving towards the opposite pole, right? I've told you it millions of times now. These are characteristics of anaphase. Remember, in metaphase 1, the paired homologous chromosome. Remember this, in metaphase 1, the paired homologous chromosome, they get aligned at the equatorial plate or the metaphase plate. Okay? In metaphase 2, the chromosome get aligned. Okay? The chromosome get aligned at the equatorial plate. In anaphase 2, the centromere divides and the sister chromatids start moving towards the opposite pole in anaphase 2. So question is asking which of the following stages of meiosis involve division of centromere? It is anaphase 2. Okay? Now it is clear to everyone? Sunita says option C. Very good, Sunita. Deepak says anaphase 2. Very good, Deepak. Now moving to the next one. Centriole undergoes duplication during S phase, prophase, metaphase, and G2 phase. Quickly answer. Centriole undergo duplication during. This question came in NEET 2021. In the starting only, when I was discussing the mitosis, I told you. And when we were discussing the interphase also, I told you this point, the centriole undergo duplication during. Deepak says 1. Okay. What about other? Ritu, MS Gauri, Lakshmi, Shreya? Sunita says option A. Okay. Very good, Sunita and Deepak. The centriole undergo duplication during S phase. Remember, DNA duplication and the centriole or the centrosome duplication occurs during the S phase. But the centrosome start moving towards the opposite pole during prophase. Remember this point, very important. So in prophase, the duplicated centrosome, they start moving towards the opposite pole. The nuclear membrane disappear in prophase. Okay? Nucleolus disappear, cell organelles disappear, then the chromatin start undergoing condensation and compaction to form chromosome. In metaphase, the chromosome, they get aligned at the equator. In G2 phase, the protein synthesis occur, which are required for the M phase. Synthesis of those protein occur which we require for the M phase or the division phase. Moving to the next one. This question number 8 says, match the stages of meiosis in column 1 to their characteristic features in column 2. Select the correct option using the codes given below. There's a column 1 and there's column 2. Column 1 says A is packetine, B is metaphase 1, C is dikinesis, D is zygotine. Okay, column 2 says 1 is pairing of homologous chromosome, 2 is terminalization of chromosome, 3 is crossing over takes place, 4th is chromosome align at the equatorial plate. What do you think? What will be the correct option for this question? Sunita says option C. Deepak says option 3. Okay. C. Okay. Now let's discuss this. So packetine, column 1 says packetine. Packetine is the crossing over, where the crossing over takes place. I told you, recombination is a characteristic of packetine stage. Packetine is 3. I write it as A as 3. Okay. Packaging crossing over takes place. Metaphase 1. Chromosome align at the equatorial plate. B. Fourth. Dikinesis is terminalization of chiasmata. C. Second. Zygotine, pairing of homologous chromosomes, synapses. So A3, B fourth, C second, D first. Correct option is option number 3. Very good, everybody. MS Gauri, Deepak, Sunita, very good. Moving to next one. Anaphase promoting complex is a protein degradation machinery necessary for proper mitosis of animal cell. If APC or the anaphase promoting complex is a defective in a human cell, which of the following is expected to occur? Option 1 says chromosome will be fragmented. Option 2 says chromosome will not segregate. Option 3 says recombination of chromosome arms will occur. Option 4 says the chromosome will not condense. What do you think? What will be the correct option for this question? They're asking the anaphase promoting complex. Remember, the hint is given in the question itself. Try to find that hint. Deepak says option 4. Okay. Anaphase promoting complex, APC. It's a very important complex during anaphase stage. Okay. So there's a protein called cohescin. This protein is very important because it holds the sister chromatids at the centromere. This protein is responsible for holding that sister chromatid at the centromere, so that the sister chromatids are held together at the centromere. Okay. And there's a separase enzyme. And the function of this enzyme is to break this cohescin protein. So this separase enzyme is activated during anaphase. Okay, this separase enzyme is activated during anaphase, so that the cohescin, which is holding the sister chromatid, together breakdown and the sister chromatid gets separate and they start moving towards the opposite pole. But before anaphase, this separase enzyme is inactive. And there's one component which keeps this separase enzyme inactive, that is securin. Okay? Securin is a protein that keeps the separase enzyme in the inactive stage. Remember this. So during the anaphase, this APC, anaphase promoting complex, degrade this securin. Once the securin is degraded, that means the separase enzyme will get free. The separase enzyme once get free, that means now the separase enzyme is active and it can act on the cohesin proteins which are holding the sister chromatid together. Okay, now when the separase enzyme act on cohesin, the cohesin will undergo breakdown and the sister chromatid, they get separated and they start moving towards the opposite pole. So this is the main function of APC. And the question is asking, if APC is defective in a human cell, which of the following is expected to occur? If APC is defective, then the chromosome will not segregate. Segregate means separation, right? Because APC is what working? It degraded the securin, and securin is the holding the separase enzyme, keeping the separase in the inactive stage. So when this APC get defective, now the securin will remain as it is. It will not undergo degradation. That means the separase will remain as inactive. And when the separase will remain as inactive, the chromosome will not be able to get separate. So the correct option for this is option number 2. The chromosome will not segregate. I hope this is clear, Deepak, now. Okay. Thank you all for your participation. It is almost 1. We are winding up the class. I hope this class is informative and interesting. Thank you all. Have a nice day. See you in the next class.